Otherwise, you will get ketone. Aldehyde can be oxidized further to acid. Acid can be oxidized to carbon dioxide. So when you burn something, you get carbon dioxide. That is the final product of burning, which is oxidation in presence of oxygen. Without oxygen, you cannot burn anything, you know that. So, loss of electron means loss of hydrogen. Always hydrogen follow electron in biochemistry. Gain of oxygen means also loss of electron. Gain of oxygen. There are many reactions takes place in the biological system. They are oxidation reduction reaction. And small organic molecules are involved. We learned they are called coenzymes. Because enzymes sometimes by themselves cannot do the job. So they rely on some helper. And the helpers are either cofactor or coenzymes. Either way, they involved in oxidation reduction reaction. NAD plus and FAD, these are the most important biomolecules. You can buy over the counter these molecules. If someone has less energy, sometimes these molecules help, and we're going to discuss how. So we're going to see a couple of questions on the homework. Which one is the oxidized form? Which one is the reduced form? So without hydrogen, they are the oxidized form. If NAD plus is involved to oxidize, for example, alcohol to something else, what would be the final form of the NAD plus? So then NAD plus will go to NADH. Addition of hydrogen means this is the reduced state. FADU will be converted to FADH2. Addition of hydrogen means this is reduced. That means something else being oxidized. So these are called oxidizing agent. These are called reducing agent. Okay. So there are many oxidizing agents, but these two are the most important one. So they accept electron. Accepting hydrogen means accepting electron in biochemistry. You need to know that. You are going to see a couple of questions. As I said, which one is being oxidized? Which one is being reduced? So you look at hydrogen transfer. Hydrogen transfer is equivalent to electron transfer here. When the electrons are being accepted, means this species is being reduced. So there's something else being oxidized. When the hydrogens are being lost, that means this is being, if BH2, either way, if this is lost, that means oxidized. But here it is being accepting. And this is being oxidized, this is being reduced. So if something is oxidized, something else must be reduced. Because you cannot have oxidation reduction without each other. Oxidation reductions always go together. You do not have to know all the names, the detailed names. But NAD plus is called nicotinamide adenine dinucleotide. And this is the oxidized form. These are the reduced form. Again, could be confusing on the homework. Whenever you see hydrogens are there, that means that is the reduced form. Whenever you see without hydrogen, that means they are the oxidized form. So if something is reduced, that means this something else is being oxidized. So this is the FAD. FAD comes from vitamin B2. It's called riboflavin. Similar way, NAD plus comes from niacin. You can buy over the counter. It's a vitamin B3. These two are very important in the metabolic processes. Metabolism cannot happen properly if someone lacks these two. Oxidized form means FAD and NAD plus, as I said. Reduced form means FADH2 and NADH. Now notice here, FAD got 2 hydrogen, NAD got 1 hydrogen. And because of their structure, so if I look at the structure here, NAD plus. This is very important reaction. Hydrogen follow electron. Wherever hydrogen goes, electron goes. So you see, I can rearrange this one. So this one, because this got a positive charge, because of the 4 bonds, nitrogen. If I remove the bond, then I can add 1 hydrogen here. So this is hydrogen. And adding a bond means 2 electrons. So these 2 electrons are being added here to this molecule. And another H plus is being added also as a protonated. So this also can be written like NADH2 plus instead of NADH with H plus. Many ways the molecules can be written depending on what form, at what pH they are. Similarly, FAD, double bonded nitrogen, 2 nitrogen. Each nitrogen has a non-bonding electron pair. They can accept the non-bonding electron pair. As a result, they can have 2 hydrogen. So the 2 hydrogens are being added. So this is called reduced form, FADH2. So I'm showing many different ways, but only thing you have to memorize what is FADH2, what is FAD, what is NADH or NADH2 plus, or what is NAD plus. Okay? You do not have to memorize the structure. But you know these are the oxidizing agent, these are the reducing agent. And that we're going to apply in next few chapters. So this is just the beginning. So we're going to see question like that. Malate has been converted to oxaloacetate. Remember, oxaloacetate. So why oxaloacetate? Because there are 2. This is acetate and this is oxalate. So what type of reaction is this? Who is being oxidized? Who is being reduced? You see, 2 hydrogen. They are being removed here to go to carbonyl. So the removal of hydrogen means oxidation. So who is the oxidizing agent? NAD plus. And what is the reduced form of the NAD plus? NADH or NADH2 plus. The same thing. It depends on the number of protons. So this reaction is called dehydrogenation, which is also oxidation. Dehydrogenation means removal of hydrogen, which is equivalent to oxidation. So the atoms are removed, so they are the electron carrier. If you follow the hydrogen atoms, you can follow the electron. And that's what we're going to do for the biochemical process. So 2H plus along with 2 electron plus, electron minus. E minus is represented the electron. So this always you will see they are together. Especially when you talk about electron transport chain. This is very important. And you have a couple of problems on the homework. So can you answer which one is the most reduced state in the 4 following molecules? Most of you answered A. What do you think? That is correct. So most reduced state. So as you go down, you can see they are being oxidized. As you go down, they are being oxidized. So alcohol first oxidized to aldehyde. It further oxidized to acid, and the final oxidized product is carbon dioxide. As I said, when you burn something, you get the ultimate product, which is carbon dioxide. Hydrocarbons are burned. So, carbon dioxide, this one has the oxidation number +4, this one has -4. So, this is the most reduced state. Now, we'll talk about the common metabolic pathway, the last 2 stages of metabolism. The first stage is, third step is citric acid cycle. So when you consume food, no matter what type of food you consume, you ended up with 2 carbon containing compound. That is called acetyl-CoA. How? That we're going to learn in next 2 chapters. So once you get 2 carbon containing compound, that will enter into a cycle that is called citric acid cycle, which has 8 steps. Citric acid cycle. Within the citric acid cycle, 2 carbons are oxidized to form carbon dioxide that we exhale. Then it produces 4 reduced coenzymes. So what are the reduced coenzymes? FADH2 and NADH. 3 NADH and 1 FADH2. I'm going to show you. These molecules will enter into the last stage, that is called electron transport chain. You need to memorize the steps, and I'm going to repeat this in next 3-4 chapters because this is the common metabolic pathway. So after the citric acid cycle, the molecules which are called electron carrier, they are the reduced coenzyme NADH and FADH2 will enter into the electron transport chain. That is the stage 4, where the biosynthesis of ATP will take place. Okay? Many compounds. So, digestion, acetyl-CoA formation, citric acid cycle, and then ATP production in the electron transport chain. So, our focus right now is this one, citric acid cycle. Why it is called citric acid cycle? How it is being controlled? It is extremely important that we need to have a control mechanism in every place. We need to have right amount of energy in right place. We don't want to have too much energy in one place. That is going to cause organ damage. So they are beautifully controlled by different enzymes. Here, one of the enzymes is very important that controls the citric acid cycle. Remember, allostatic mechanism that can take place to turn on or to turn off a system. So here the system is citric acid cycle. So now our focus is to know what happens in the citric acid cycle and why and how it is controlled. So can you tell where does the citric acid cycle take place? Nucleus, ribosome, Golgi apparatus, cellular, mitochondria. We discussed this yesterday. So next few days we're going to do a little bit different way. So I'm going to ask you more questions. The steps are important. You need to connect them. So you know the flow of energy, flow of thought, everything is beautifully matched. And you're going to see the result. %, very good. Don't forget this. So, citric acid cycle. What is the purpose of citric acid cycle? That is your goal. What are the requirements for the citric acid cycle? And what are the products? So the starting compound is 2 carbon containing compound that is called acetyl-CoA. 2 carbon will get into the citric acid cycle. So the first one has 6 carbon and the name is citric acid, and that's why it is called citric acid cycle. So citrate or citric acid. Then it will go through a series of steps. Some steps, so this is step number 1, 2, 3, 4, 5, 6, 7, 8. 8 steps. Each step requires something. So step number 3 is the most important step that can turn off or on by enzyme that is called isocitrate dehydrogenase, which I'm going to show you in next slide. You are going to see a question like, what is the first step in the citric acid cycle? So, formation of citric acid. This is also called Krebs cycle. So scientist Krebs that invented this cycle. This is also called CAC cycle, citric acid CAC, or TAC, triacetic acid. So you have 3 carboxylic acid group, 3, and you have total 6 carbon. So how many reduced coenzymes are formed? 1, 2, 3, 4. What are they? So they are NADH, 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 3 NADH, and this is FADH2. These are very important to know the numbers to calculate how do you get 32 from 1 glucose unit. Okay? So that we are going to discuss later. How do you get 32? So the numbers are very important. 1, 2, 3, and 4. 4 reduced coenzyme. Where do the oxidation reduction takes place? So you see the reduced coenzyme means these steps are the oxidation reduction steps. This can be divided into 2 halves. And we're going to, one is called generation, another is called degeneration. So what is GTP? Now you notice there is one GTP here. One GTP. GTP is equivalent to ATP. But for some reason, cells prefer ATP, the energy from the ATP. So no one knows why the cell's currency for energy is ATP. So it's like someone producing, if you think about like dollar, Canadian dollar and US dollar. You cannot use Canadian dollar in USA. So that has to be converted to US dollar. Exactly the same way, if the cell somehow in some places GTP is produced, GTP must be converted to ATP before cell can use that energy. And I'm going to give you several examples to this analogy. Again, you are going to see question, does ATP, is there any ATP production in the citric acid cycle? No. Is there any energy production? Yes. So that energy is different than the ATP. So what is GTP? GTP is guanine triphosphate instead of adenosine triphosphate. So only that one molecule is different, one compound attached to this. It is just like ATP, but it must be converted. So how it is converted? So the energy from the GTP is transferred to ATP, then ATP can be converted to ADP. So again, ADP means diphosphate. Another phosphate is transferred from GTP to ADP to make ATP. So like making GTP is ultimately like ATP, but the cells prefer energy from ATP. So GTP got to be converted to ATP. This is important. So transferring phosphate from one molecule to another molecule is called substrate level phosphorylation. So we're going to see, and this is very important in next few chapters, what are the differences between substrate level phosphorylation and oxidative phosphorylation, which we are going to discuss tomorrow in stage 4. So substrate level means it involves a phosphate transfer without any electron transfer, no oxidation reduction reaction. But oxidative phosphorylation involves electron transfer, which is the last stage, stage number 4, where the ATP production takes place. So you can see that these 8 steps can be divided into 2 halves. This is called generation path. The first one is called generation path. And this is called regeneration path. So that means the energy is released by these steps, energy required by these steps. Okay? So you have a question on the master in chemistry. Which one? So step 1 to 4 is called generation. Step 5 to 8 is called regeneration path. Now, can you answer? Does any step of the citric acid cycle directly produce ATP? Explain. A or B? Which one is correct? Most of you answered A, and that is correct. So step 5 produces GTP, which is not ATP, but the energy content is the same. So the GTP is going to transfer a phosphate to ADP, which will be converted to ATP. That can be reused. So energy production is there, but no, there is no direct ATP formation here. So if I look at all the steps, this is a little bit complex, but we're going to simplify later on. We're going to discuss, we already told you that investment is required to gain more ATP. Why the investment is required in the first step, we'll discuss later. That is not important for this class. But what you need to know for your homework problem, you're going to see the problem. Where do the oxidation reduction takes place in the citric acid cycle? So the answers are going to be step 3, step number 3, step number 4, step number 6, step number 8. Oxidation reduction takes place. Where do the carboxylation reaction takes place? Step number 3, step number 4. Decarboxylation reaction. So decarboxylation means production of carbon dioxide. That carbon dioxide we exhale. As I said, we sometimes have misconception that oxygen we inhale, that oxygen combined with carbon to make CO2 that we exhale, but that is not true. This carbon dioxide comes from the 2 carbon in the acetyl-CoA, and the oxygen we breathe in, that is going to be used to trap electrons and H plus to make water. That we're going to discuss tomorrow. So in every step, depending on our body need, we need different amount of energy in different places at different time. During exercise, you need more energy. During sleep, you need less energy. So how these processes are controlled? And who controls? Enzyme. What is the name of the enzyme? Isocitrate dehydrogenase. In which step? Step number 3. It is going to take some time, but the good thing is that because you are going to repeat this again and again, you should not have any problem at the end. After we discuss 3-4 more chapters. Okay? So this is very important. Isocitrate dehydrogenase that controls when no more energy required during sleep. You need less energy. So then citric acid cycle must be slowed down. How it slowed down? Again, allostatic mechanism. So we're going to discuss how it is slowed down in a few minutes. So how do you account all this energy? We're going to discuss in later. But this is the resulting reactions of citric acid cycle. You start with 2-acetyl-CoA. You used oxidizing agent, NAD+, FAD, and GDP. That will be reduced to NADH, FADH2, and GTP, which is the energy. So this is the resulting reaction. And the carbon dioxide we exhale, that comes from the citric acid cycle. Do not forget that. The carbon dioxide we exhale, that is the product from the citric acid cycle. The oxygen we inhale, that is required in the last stage, which is called electron transport chain. And I will repeat this again. So you should not, and always you expect question, where does the carbon dioxide form? Where does the oxygen require? So the production of these reduced coenzymes are essential for the last stages, which is called electron transport chain. That is the energy production step. Okay? So regulation is necessary depending on body need. We must have some control mechanism. We don't want to have excessive energy in one place or too little energy. It must be regulated depending on our body need. We notice when you exercise, we breathe harder and harder. Why? That we're going to discuss in next chapter. But right now, you need to know citric acid cycle must be controlled depending on our energy need. So isocitrate dehydrogenase plays a major role to turn on or turn off this step, which is step number 3. How it is turned on? Remember, ATP is the final energy molecule. ATP, when ATP is used, then you have more ADP. More ADP means low energy. So when low energy, then this is going to bind to the enzyme allosterically to activate. Then citric acid cycle will turn on to have more ATP production. But again, ATP production in the last stage. So before that, you need to have more reduced coenzyme. So these are NADH and FADH. When you have too much energy, then you have too many NADH and FADH2. Then they are going to act like an inhibitor. That means they are going to slow down or stop this process. So citric acid cycle is going to slow down. So this must be controlled. And the isosociated dehydrogenase is responsible for controlling the rate of this reaction. Can you answer this question? So what is the purpose of the citric acid cycle? Most of you, 92, very good. That is the correct answer. So that's all for today. Tomorrow we're going to continue.